Hi everyone, welcome back. It is AP Bio time. I am excited to be here with you. We're going to cover some pretty exciting content on cell cycle and homeostasis. So give me a hi or a hello and let me know how you are doing in this fine Wednesday afternoon. I know the days are probably starting to blend together, aren't they? It's hard to remember. Every day I have to remember, okay, let's check my calendar. What day is it actually? Because they all seem the same. Yeah, so I mean, we got some good chatter going on in terms of comparisons between different AP courses. History is so different from the sciences, and I don't know. It really kind of, I think that's like the hard dividing line between your interest more than anything else. Yeah, so the AP Bio questions are difficult. I'm not going to disagree, Aeri. um, and it is interesting. Hey, welcome back, Alvarin. I'm excited to see. I am tired too. I hear you. Yeah, please do spread the word. We're excited to do more. If you are interested in doing more of these or more in a different test subject, you got to spread the word. 안녕하세요. Hey, look at that. Yeah, and you see purple? Not quite yet, but you should see it soon. So let's get going. Let's get going. Okay, now do you see purple? Now do you see my big purple screen? I know it's we've changed some things around. It looks very jazzy. It's very bright. But yeah, once you get there, let me know, and then we will get started with our AP Bio class for today. Now, it's not a class. We're not going to review the science, although we might review some content in the context of doing some free response questions together. Jason, thank you. So yay, yeah, I know it has changed, right? Yeah, okay, so there we go. Now it's purple, right? Okay, so if you have not had a chance to tune into these before, we do have them all posted on the YouTube page too, so do plan to check those out later on. Just a brief, quick mention of what we've been just, talking about, the info that we know we are going to share with you, right? It is 45 minutes, it is only free response. It's not going to test last 20% of the classes. CollegeBoard is offering some free online classes. I do believe they actually start today for the students who do want to kind of complete their education. For the AP Bio test, it's going to be ecology and evolution kind of stuff. That's going to be there. So I like it. You're right. It's about 400 nanometers. There will be 2 tests. I believe the one is still planned for May, but we don't really know what the dates are. It's not going to be limited by technology. So if you want to write it on paper and take a picture, that is going to be allowable. But more information about the number of questions, the question types on each exam, all of that will be posted on the website at April 3rd. So yeah. We're doing the best we can. We're making some educated guesses as to what will be happening. At least we do know it is free response, and therefore you can start like customizing your studies towards what is most likely to show up on the test. Okay, so we're going to do our best as well. We're going to do our best. Okay, so let's see, what I thought would be happening, would be good, is yesterday, we had a couple of questions about like, what were some of the requirements behind the task verbs, the ones that are bolded in the prompts. So I grabbed a couple of them and reworded them, and I threw them on here. You can always come on and flip back to that. But this entire list of verbs is also on the AP the bio guide, the huge PDF that the AP Bio it has that is released from the College Board. So do check that out. It's near the end of that giant document. So there's a whole page of these with more descriptions on like what these verbs actually require you to do, so that you make sure that you get all the points you can. Isabella, the College Board videos are on the College Board site. So if you just Google that, you should see directions on how to get to those. Yeah, they do have them. Yeah. So in particular, yesterday we're like, what's the difference between identify and describe? Well, identify is just, you don't need additional information. You just got to state the fact. Whereas describe, you need more. You need to fill in details for whatever that topic is. And so yeah, take a look at some of these maybe a little bit later on. You can always rewatch these videos later on and see if your answer choices are fulfilling the requirements described by the task verbs in the question. So now I'm going to give you a chance to do some of this with some pretty cool and interesting problems we're going to talk about today. Now, I know biosystems is not tested on AP Bio anymore, but you could still be describing them in a stimulus. Like you could still get an entire stimulus around biosystems. It's just not, what it means by not being testable is that you're not required to have that memorized and know that as fact for the AP Bio exam. So that's just context for the first stimulus I'm going to have up here. And yeah, Ria, how often do we do? We're going to do, so today, Thursday and Friday. Friday technically is the end of our current sprint of these videos. If you want more, you gotta ask for them. So definitely like come back, tell your friends, comment, things like that, so that we get enough incentive to do more of these. I really do hope we get to do more though, and because these are kind of fun. Okay. Okay, so just like all the other times, I'm going to give you some time to kind of work through this on your own. What I would love for you to do, just like yesterday, is type in your responses, but don't hit enter into the chat. And then once I tell you to go, hit enter, we're going to see all the answers together so we don't spoil it for really anyone else, okay? So let's do that. I'm going to give you 3 minutes, like usual, to consider the stimulus and the first question. And then there's another part, and we'll give you additional time for that second part. So here we go. Here is the first one, 3 minutes to answer the question for A. You can use the diagram on the side to help you out. And so remember what describe means, okay? So 3 minutes for this prompt on your own. Type it in, but don't hit enter. Okay, come on back and tell me what you thought about estrogen's role in this feedback portion of the menstrual cycle. Go ahead and hit enter into the chat, and I'd love to see what you guys all came up with. So estrogen is important in the regulation of the menstrual cycle in women. We do have it on days 5 through 9. Talk about how the follicle, as it grows, will secrete estrogen. Then estrogen then travels to the pituitary gland, and that decreases the amount of a particular hormone. Well done. Look at that. I love this. This is so fun. And I'm so encouraged to see so much more active participation as we go. Nice. You're right. Well done. Okay, so remember, key points for free response questions. You cannot put them in bullets. You need full complete sentences. It's okay to make them short. They don't need to be fancy, but you do want to have complete sentences and as correct spelling as much as you can. So estrogen is positive feedback because something. So it's a continually release until ovulation occurs. Nice. Interesting. Okay, so remember that because biosystems is not testable, you want to make sure that you're using details from the stimulus in order to support your idea of why, what is feedback, what is an example of the negative and positive feedback. Nice. Okay, so I like this. Elise says, estrogen works as positive feedback on day 10 when levels are high, because estrogen travels to the pituitary gland to increase the amount of LH. It increases LH, and then that is positive feedback. That is exactly correct. So anything that is going backwards, but increasing what's going on previously is going to essentially build up like a cascade of some sort. And Sadie says estrogen says something about the positive feedback. Nice, stimulating ovulation. This is what we call the LH surge. It is absolutely required for you to have high amounts of estrogen, which is going to induce that big surge of the LH hormone, and then that's going to luteinizing hormone, LH hormone, this luteinizing hormone. The LH in order for you to see that ovulation event occurring. Okay. Nice. Okay, let me see if I can find an example of the negative feedback from the chat. You don't know what I will do, says estrogen works in a negative feedback loop by inhibiting the production of the follicle stimulating hormone, which is a hormone that signals the production. Okay, so here's the thing that you put in the brackets. You want to, again, be very clear, because that's not what the hormone says or is defined as in the stimulus. The stimulus does say that follicle stimulating hormone is what causes the follicles to mature. So it can negate itself, like some of the points that are given if you state a wrong fact, whether it is like content that you need to know, or it is something that is based on the details that are provided in the stimulus. If you state them incorrectly, it is very possible they might take away that point. So be very careful about how you go about in choosing to support your ideas and your claims. So if you want a description of that, let me just throw that in here as well. So a proper kind of a sample response here would be something like this. Cite the days, talk about the type of feedback, and say why it is a negative or a positive feedback by using the details in the prompt. So estrogen does have this really cool role where time kind of does both depending on the day that you're in. And so that's cool. That's cool. This is how the menstrual cycle works. So here's the thing, Kaylin. If it said describe how estrogen acts in one of, like if it said specifically one, then you pick one of the two. That's fine. But since it doesn't say specifically one, it does say both, you need to do both. Yeah. So the question stem is going to give you the information, the requirements for the answer choice. What happens if it says just do one and you do both? Do you guys all know what happens? What do the graders actually end up doing? I read up on this because I was very curious as to like, how did they decide, like if they give you both and you're like kind of playing your chances, you're gonna give as much information as you can. Yeah, if it says just do one and you do everything, they're only gonna read the top one. They're not gonna, they've got a lot of papers to read through, so they're not gonna sift through and find the one that looks the best. They're just gonna read the one right at the top. So all it's gonna do is just eat up time. So don't do it. Just pick the one that you think is going to work the best for you, describe it, and then move on. Don't do both. They read the first one for you, got it. You don't lose the point. You won't lose a point by doing more unless it contradicts the first one, but it will only read the top one. Yeah. Okay, nice job, team. Nice job. Okay, so we're going to use the same stimulus, and then we're going to see like a variation of this, and it's super cool. Okay, so I'm going to go ahead, and this one I'm going to give you 2 minutes. It is kind of a tougher problem. It is using the same stimulus, but it's going to take you some time to kind of work it through. Feel free in your scratch work. I know you have to write it down, so in text form for the answer, but feel free to use some diagrams to help you figure this out, okay? So let's take 2 minutes to do the next prompt. Go ahead and type in your responses, but don't hit enter until I tell you to go. So 2 minutes for this next one. Okay, there you go. Okay, so this is your chance to shine. Go ahead and type in enter and tell me, what did you think about what's going on? What can you predict about what happens when you take Clomid, given the information and the stimulus, as well as information about what is going on in chronic anovulation? I'm gonna be honest, team. I don't know who Ryan Barrow is. Maybe that comment was for someone else. But if it is somebody that I do know, then I apologize for that. Hello to Ryan Barrow out there. Okay, nice, well done. It promotes fertility. I mean, that kind of makes sense, right? If it is a treatment for fertility, tell me how. How does this promote fertility? Okay, but not bind, decreases amount of fall. Ooh, let's think about this. Let's think about this. Okay. This one isn't as clear cut, I will say. It's okay, but it's still within the framework of homeostasis, about feedback mechanisms, okay? So let's think. Okay. So here's something interesting. In order for feedback to occur, whoa. Hi, everyone. Sorry for the tech issues. I'm so sorry. But hence, you know, the trials and tribulations of online learning. So I'm just reorienting myself. I had to restart my entire computer. It's good times, team. Hey, yeah, I know. So, okay, can you guys see and hear me okay now? Let me know, and then we will get started right back on. Okay. Yay! Okay, we're back. Okay, let's do this. I know, of course, technology. Thanks. I know, but like so convenient, really. We're blessed. We're blessed with that technology. When it goes wrong, does it ever go wrong? Okay. Okay, let's do this. Let's do this. I'm just pulling up my notes 1 second. I have to rearrange my screen. Okay. Okay, so I'm not entirely sure where Nick left off, but you guys all had some excellent answers about clomid. So what I'm going to do is, instead of assuming that you don't know the answer to this, I'm going to quickly just recap what is going on in this part. And then we'll just kind of move on, okay? So chronic anovulation is a condition. They don't have enough ovulation to occur. And what you want to make sure you understand, where's my pen, is this component here, right? So receptors are things, good, okay. So receptors are things that are essentially like, yeah, I always imagine these as like little cups. And so this is like a signaling molecule. In order for estrogen to go and feed back on something, it needs to have a receptor there. Okay, so think about this. Estrogen goes backwards into the pathway. So this is my little estrogen, I'm going to call it E molecule, and it binds to an estrogen receptor that exists on the pituitary gland. So normally this estrogen receptor, at least in the context of, let's say, follicle stimulating hormone, this guy normally goes back, and then what happens is that you get a negative feedback event occurring. So normally this is negative feedback, but if you have something like clomid that essentially blocks that estrogen receptor, you block the way, then essentially you get no negative feedback occurring. No negative feedback occurring, which means that you're still going to get lots and lots of follicle stimulating hormone, which then goes and induces more and more maturation of additional eggs. So here's a fun fact. Here's a fun fact. One of the unintended consequences of having clomid, very popular these days of fertility treatment, is a certain kind of birth. And I would love to hear from you. What do you think? What is likely a not expected, not intended, but an expected consequence of having a treatment that increases the amount of follicle stimulating hormone, which leads to more and more follicles maturing? What do you think? It's a cool link, and this is how feedback is working. This is how we use drugs to disrupt homeostasis, disrupt feedback mechanisms. So if you're thinking, well, if I block that and I mature multiple eggs and multiple eggs then get fertilized, I will likely have more and more multiple births. And that is absolutely true. We see a lot of multiple births. Yeah, it's not quite ectopic. Remember, ectopic pregnancies are essentially when the egg is implanting in a place that is not in, where it's supposed to be. But yeah, twins and triplets, Sadie, got it. Yeah, I mean, it could lead to prematurity. That's not an unusual kind of thing with multiple births, but really the consequence of a lot of this is that you get a lot of babies, and they're fraternal children, fraternal multiples, not identical. Nice. Yeah, it is going to result in higher fertility. No, the intention behind this isn't necessarily like they're trying to induce multiple births, but if women are suffering from a condition where they're just not ovulating at all, then increasing the amount of hormone is likely to result in at least one follicle successfully maturing and being giving that. Yeah, Flora, it's not the only reason, but it is one of the things. This clomid is like one of the first line of treatments that women usually will have. And it's very specific. Like you have to take it on very specific days in order for it to work. Because if you take it on the wrong days, it's not going to work properly. Okay, great question. Is there a limit on how far we should go when predicting? Because the answer stated more than one egg can be fertilized, but I said that there will be more than the usual amounts of maturing follicles. That's a great question. Here's the thing. This is for women and this for humans. And while I feel like it's hedging on the line of expected knowledge versus things that is not quite testable, how many babies are we supposed to have? Like humans are supposed to carry, like, is it weird or unusual or like an at-risk kind of pregnancy when it's multiples, or is it that, it's expected and it's fine and you're going to progress us forward? You can't really decide, at least with clomid, Isabella. You can't. Like if you do in vitro fertilization, you know, that will implant a certain number of fertilized zygotes, right? And then it's a guesswork kind of still there, but you can't control this. We don't know how many of these follicles are actually going to end up maturing. So so the answer, yeah, twins are common, I agree, but you know, since we are, as humans, are meant to really just carry one baby at a time, we're not really meant to have like litters and stuff like that. Yeah, exactly. Yeah, you were usually meant to just have one. So the thing to kind of think about is that the more than the usual amount of follicles is correct if you think, okay, the usual amount is just going to be one. That would be fine. Okay. Yeah. So if you're at all like sitting in the situation where you were like writing out your answer and you're like, I don't know if this is like a bit much, hedge it. Like put some moderating statements around it. At least it'll give you a little bit more wiggle room for interpretation, at least for the examiners. Okay. No, Flora, identicals tend to, I think the only influence with identicals are genetics. Like, I don't really know if there's any ways to induce the splitting of a fertilized zygote. But yeah, fraternals are like drugs like this are how you get fraternal twins. Nice, okay. Okay, so let's move on. We got a couple more questions here. I just need to find my timer. There we go. Okay, so let's go on. And so here's some more like bigger picture basics. We're going to do a little bit of cell cycle review here, but just like before, I want you to take 3 minutes to analyze the stimulus, which is quite short, but you have to do a lot for this stimulus. So 3 minutes to write down 3 differences between mitosis and meiosis. I'll see you back in 3 minutes. Okay, now go tell me some of those pertinent differences between mitosis and meiosis. What are those 3 differences? There are a number of things that you can type here. So it is pretty broad and big, and you can be pretty, you can make use of the content you do now. So I like this. Alicia, 1 division, meiosis has 2. Yeah, so there's 2 rounds of division. You got it. Mitosis results in 2 daughters identical to parent. Meiosis results in 4 non-identical haploids. Nice. No, mitosis occurs in somatic. Flora says meiosis occurs in germline cells. Per growth and repair. Oh, I love the purpose part, Elisa. Yeah. Yeah, well done. You guys know your stuff. Jason says 2 identical diploid, haploid. Oh, yeah. Okay. No, so here's the thing. Diploid, haploid, those are not human-specific terms. What makes it human-specific is the number of chromosomes, because generally speaking, the division still kind of happens the same sort of way in general, right? And Naya says mitosis is PMAT1, so meiosis is PMAT twice. Yeah, I like that. I've got that in my notes as well, actually. Genetically identical versus genetically varying. Got it. Okay, okay. And for the person who was asking a little bit earlier, if we're going to go a little bit later, I just got permission to do so. So we can just go a smidge over. Okay. So I do apologize for the tech issues. So, okay. So let's track all this great information you guys are coming up with. Not a highlighter. I need a pen. Okay. So let's do this. Mitosis. And then we have, yeah, we'll do this meiosis, like this. Okay, so let's see this. Favorite difference. I know, I love talking about this part, I really do. So PMAT, just 1 times, right? And then PMAT, right, 2 times. Here, mitosis is 1 cellular division, meiosis is 2, you got it. The type of cells, right? So these are what we call somatic cells, or somatic cells are the kinds, I mean, by definition, they're essentially the ones that are not germline, right? So germline are like the ones that are used for sexual reproduction, and it does result in different kinds of genetic information. So 2 identical diploid. So what does diploid mean? And how haploid? What do those terms mean? 4 identical not identical. Almost got trapped there. Not identical, but yeah, so non-identical haploid cells. Yeah. Ooh, yeah, crossing over, random fertilization occurring in meiosis. Yeah, so you get a genetic variation because of those recombination events. Yeah, and so these are like what we mean by germline are the ones that are essentially going to undergo the process of spermatogenesis or oogenesis, create your eggs and your sperm. Those are the ones that are then going to be what we call gametes, and then gametes fertilize together, create your zygote. So yeah, haploid means half, diploid, di means 2. So a common way to kind of denote this, N is like the ploidy, how many like, you know, one set of chromosome is an N, okay? And so a diploid is what we would call 2N, and then a haploid is what we call N. You got it, Elise. Well done. Diploid means 2 sets, haploid means 1. Humans only really go up to that. If you go beyond those kind of ploidies, you've got like other issues kind of going on with your with the progeny. But did you know that plants have all sorts of crazy ploidies? They got like heptaploidies and octaploidies. I was reading all about it yesterday. And it actually like they induce polyploidy that way, and it creates like really huge flowers. And this is why you get like those crazy giant strawberries in the supermarket that look like the size of your hand. They don't taste so good, but they're humongous. A lot of that is done through the induction of polyploidy, actually. Yeah, Jason, polyploidy, got it. So Isabella, haploid and diploid is the number of sets of chromosomes. So if in humans you have 23 chromosomes, it's not an if, but like yes, they have 23 chromosomes. Well, N, a haploid cell, would have 23 chromosomes. Whereas a diploid cell, which is essentially pretty much all the cells in our body other than the eggs and the sperm, are going to be diploid, and that would be 46 chromosomes. Yeah, decaploid. I know I went down this giant rabbit hole yesterday about polyploids. It's really interesting, actually, really interesting. Let's see. We can talk about the genetic variation. So no genetic variation, right? Genetic variability. See, this would be like due to the whole crossing over events and things like that. Here's one. When do sister chromatids line up at the metaphase plate? Ephraim, yeah, 23 is haploid in humans, yeah. When do sister chromatones line up on the metaphase plate? I kind of gave it away, but that's okay. Sister chromatids. Exactly, yeah. Say Vishnu, recombination only occurs in meiosis. Yeah, you don't get any recombination happening in mitosis because mitosis is like carbon copies. Like, yeah. So here, let's make sure. Jason, you're on the right track, but I want you to make sure you're answering the correct answer. So what part of mitosis do we get sister chromatids lining up at the plate? So you're right, it does happen in mitosis and meiosis too, Holly, happen. But yeah, this is just metaphase. There you go. Good, okay. Whereas sister chromatids line up at, and then its Viv says metaphase 2. Got it. Okay, so there's only one metaphase in mitosis, so we don't have the 1 or 2 prefix, but then you gotta make sure what's lining up at metaphase 1 when we're doing meiosis then. Middle, nice mnemonic, Elise, I like that. What's lining up when you've got at metaphase 1 for meiosis? Well done, Ali, well done, Janaea, nice recalls, team. Yeah. So at metaphase 1 of meiosis, you don't have sister chromatids that line up. They are homologous chromosomes, So it's essentially, Jason, you got it. It's like, chromosome, 2 from mom and chromosome 2 from dad. That's what we call a homologous chromosomes. So they line up together side by side like soldiers, and then they kind of like split apart when the cell splits over. Yeah, so it's not just chromosomes, but they are homologous chromosomes that do line up. And that's usually where you get the recombination event happening. Good, well done, recall. Now, clearly you should not do it like this when you are writing down for a free response question. I did it for the purpose of just a quick review, but you do want nice, like long, complete sentences for your 3 differences between mitosis and meiosis. Nice recall, team. Okay. So this was kind of hinted, somebody kind of hinted in this direction for the next part. I'm not going to give you as long, but let's go ahead and take a look at the next question here. We're looking for an abnormal consequence of disrupting mitosis, and you need 2 things, and another abnormal consequence of disrupting meiosis. So I'm going to give you 2 minutes for this one. Go ahead and type in your answers, but don't press enter, and we'll review the things that you come up with together. Okay, everyone, go ahead and hit enter and tell me what is something that goes wrong when you disrupt mitosis and meiosis. What goes wrong? Sometimes the way to talk about abnormal circumstances behind processes is to think, what is its intent? What is its purpose? And then to think, okay, well, if I'm going to break that, well, the consequence will be the opposite of its purpose. That's what I would think. So Holly Happen says abnormal mitosis leads to unregulated division, form tumors. That's exactly it. Meiosis can lead to genetic deformities. Ooh. Yeah, so we can elaborate just a little bit more, I think. So what kind of genetic abnormalities are we talking about? Like, not the kind of disease, but rather like, how is the genetics disrupted, I guess. Apoptosis is another way to think about it, Michael. I like that too. Mutations, what kind of mutations? So be specific, be specific. The more specific you can when you're elaborating on content, the more points you're gonna get. You know, somebody a little bit earlier, just before I came back, was asking about things like, stated a really good fact about disjunction. And that is actually directly related to what we're talking about here, because one of the ways that you can disrupt meiosis is by having inappropriate separation of chromatids and homologous chromosomes. And that's what we call non-disjunction. Non-disjunction just means that you don't have separations of chromosomes properly. So Sadie, exactly, things like Down syndrome, which is a trisomy 21, so abnormal number of chromosomes, that's what we call aneuploidy. You don't need to know that, but you could just state abnormal number of chromosomes. Yeah, you got a trisomy 21, the most common ones of the trisomies out there. Cells might not fully develop. Yeah, that makes sense. Resulting daughter cells, genetic mutations. I like it, okay. Could they be bar buddy? Bar bodies. I said bar buddies. Bar bodies. So here's the thing, that's such a good one, Mia. It's not that they would all form into bar bodies, but it's likely that you're gonna get eggs that's going to have either completely empty or they're going to have too much genetic information. So this is still another type of aneuploidy. Yeah, non-disjunction is kind of what I was looking for, but if you said other elaborations where you just describe that, I think that would still work. So here are some things to keep in mind for the answers. Yeah, it's not intersex. Those are types of trisomies. And if you're going to get a trisomy beyond 21, the ones that are still survivable are the sex chromosomes. The sex chromosomes actually allow for a fair number of aneuploidy and still be like something that you can survive and live with. Bar bodies are actually what is left over in the process of oogenesis. So I wish I had time to go through it because they are actually super interesting. But if you think about this, I wrote down mitosis, and then let me write down the one for meiosis. But the purpose of meiosis is to essentially make it so that you have how much genetic information. I'll talk about the bar bodies in just a second, actually. Yeah. Nice, they are discarded, but why are they discarded? If you think about how meiosis work, let's elaborate on this. Actually, this is pretty an interesting topic. If I can get, there we go. Okay, so let's think about this. If I have meiosis, right, and I have, I'm gonna draw 2 chromosomes. I drew this too small. Yeah, polar bodies are the same thing, Jason. Yeah, polar bodies. Yeah, no, you don't need to have specific issues, Rhea. What I would be looking for is something that shows you that you understand that there's an abnormal number of chromosomes. Oogenesis is making eggs, yeah. They're leftovers after meiosis. That's what bar bodies are. And so this splits, right? And so I get 2 cells. This is meiosis 1. And so I get, and pretend I've got like way more chromosomes in here. Okay, and then this also splits into 2. The reason why you actually go through meiosis, team, is to get rid of leftover genetic information. Because if you fertilized a diploid cell with another diploid cell, you have an abnormal number of genetic information. So the process of meiosis is actually done so that you get to a place where you have the number of genetic information. But eggs are like, if you've ever seen a chicken egg, it looks not like a chicken egg, but it's got the same idea. It's got the yolk in the middle, which is all the important stuff, and then it's got a bunch of nutrients around it. The reason why oogenesis works the way it does is that it needs to split, right, to have the number of genetic information, but it wants to keep all the stuff. So it keeps all the organelles and the nutrients and the stuff around it. And it wants to throw away the excess genetic information. It doesn't quite have enough to split it evenly and form 4 complete eggs, and that'll still develop properly. Yeah, barred bodies are supposed to happen, but what happens in spermatogenesis, you get 4 equal. Sperms are okay, they're simple and they don't have a lot around them, but the eggs need a lot of nutrients around them for them to grow and get bigger. And so in the process of oogenesis, you only really form 1 egg for the meiosis process. Okay, so even though technically you form 4, between 3 and 4, depends on if you get a second meiosis for the first split, you only really form 1 egg per meiosis, per process of oogenesis. You got it. So it can be 3. Sometimes, you know, I've seen literature talk about 2 polar bodies, and that's it. Yeah, you got it. Like the word ooO is egg, essentially. Yeah. Oogenesis, the female version of spermatogenesis. You got it. And you'll learn a lot more about that in further future bio classes if you take them in college. Okay, so that was a fun talk. We're going to move on and do one last one, and then we're gonna call it, I believe. So let me clear this information and move on to this next one. So here I'm going to give you 2 minutes. There's not a lot of data in here, and this is pretty short and the question is quite short. So 2 minutes to work through this one on your own. Just like before, go ahead and type in your answers into the chat, and then we will talk through them together. Okay, team, here's the thing. I wasn't going to say anything, and I don't share your handles on YouTube. It is not safe, so please don't do this. Don't share any of your personal information on a YouTube stream. Let's focus on this, and yeah. Let's focus on this question. So I love the answers I do see coming in. Go ahead and type them in. The research question, what did they have in mind as they were formulating and going through the process of constructing this experiment and gathering the data? What is that all about? I love how specific this is. That's it. Like, that's the true scientist. It's not being vague and general. You want to use the specifics that are provided for you. I like it. You know, if you looked at this, it looked at the table and like the dependent variable is this and the independent variable is this, and then you kind of fuse them together into a sentence, that would essentially be a really good research question. So if you were a little bit stuck on constructing something, that's how I would go about it. Nice. I like it. Okay, look at this. So one of some of the things that I thought of maybe two variations, but I'm gonna be honest, this is a fairly straightforward piece of data. There's nothing really all that complex. And I can only really come up with two. There's possible that there's more variations, but what is the effect of insecticide on the growth of the parsnip? What phase of the cell cycle will be affected? Those would be both legitimate kind of questions that would be answered by the data here. Beyond that though, there's not a whole lot else, I would say. So Kyla says, does the solid concentration of the insecticide have an effect on the cell division? Nice, that is great. And it fits with this. We're not assessing how like the different concentrations, but that specific concentration, is it going to have that effect? Yeah, well done. Does the insecticide contain chemicals that inhibit mitosis? Guys, I'm so impressed. You came up with way more variations. I was talking about this with Nick earlier. We're like, we can really come up with two. So awesome job. I'm impressed. I'm impressed. Like, you got it. Dwyer, nice. Nice. I like it. Okay. So let's move on. Let's move on. Let's do another one then. Okay. So here is another prompt for the same stimulus. I'm going to give you 1.5 minutes for this one, 1.5 minutes to go through this next prompt, and then we'll talk about it together. Okay, everyone, throw in your answers. Researchers have said the above insecticide is effective. Using the information, evaluate and tell me, would you support this claim about whether or not this insecticide is effective? Nick and I were talking about this while you guys were working on this question. We're like, what is this? What is a pause? Interesting. Let me ask you this. Let me ask you, this one's a little bit of a tougher one. Tell me, what is the purpose of an insecticide? What is the purpose? You can't just say support. You need to provide some proof. Yes, Holly Happen has exactly what I'm looking for. Insecticides are for killing insects. It's a pesticide, right? So he's saying above, oh yeah, based on this, insecticides totally work. I mean, they definitely have an effect on the parsnip root, but is that the intended effect of an insecticide? No, it's to kill insects. You got it. It's to kill bugs. It's not actually to help, plants grow. It's to kill the pests. And that is not within the scope of this experiment. We're really just assessing, based on the first prompt, whether or not the insecticide has, deleterious negative effects on the plants that they're trying to protect it from. So that's essentially what I would be looking for. It's a little bit of a trap, it is, but you got to be careful here. Just because it does something does not mean it is effective. Okay, just because it does something doesn't mean it's effective. Insecticides are using it to kill insects. They are not. I see. Yeah. Support the claim. Insecticides are proven. Be careful. Be careful. Yeah, but here's the thing. It's just because it does something doesn't mean it is effective. That is not the intention of an insecticide. Yeah. Yeah, I mean, what is the research question? They're trying to figure out if they can use this insecticide and protect the plant from being eaten up by bugs without affecting the actual plant itself. Like, that's really what they're going for. That's why they're doing this kind of side research, right? But if it turns out that your insecticide does something bad to the plants, then you got to ditch it. It's not going to work, right? It's going to do just something as bad as the bugs just eating the plants around. Yeah, it's not that the data is there. It's just the study design doesn't address it. Yeah, it does affect cell cycle, but that's not the intent of an insecticide, team. Insecticides are there to kill off bugs. It is there to kill off the bugs. That is not the intent of the insecticide. Okay, we got one last one to go, and then we'll wrap it up for today. Okay. There we go. One last one to go. So 1.5 minutes again for this last question, and then we'll call it a day. Okay, go ahead and throw in your answers, team. No handles, no identifying information, team, in the chat, please. Okay. I like where you're going with that, Alexandra. So Alexandra says the heavier root would be the one with the more cells. Since the one without insecticide has more cells in mitosis, then it would likely be heavier because more cells are being made. Exactly it. So take a look. You're using the proof and the data to support your conclusion and your prediction. Got it. Well done. Nice. Okay. So I like a lot of the answers that you guys are putting in. Mine is very similar. I don't think it's any different. I think you got some really solid answers going in there. The parsnip root without the insecticide would likely be heavier. There's more cells going through the process of mitosis. Remember, mitosis is like just growing. It's for the purpose of division to create more and more cells. The more divisions you got, the more growth you're going to have. So it's going to be bigger and heavier. That is it. So more cells, heavier mass. You got it. Well done. That's all good. It's all good. It's all good. Well done, team. I am impressed. You guys are doing great. I know it's hard not to just recognize answer choice as a multiple choice, but to actually force yourself to write it down. But this is practice. You can get better at this. And so we are going to come back tomorrow at the same time and do transcription, translation. I'm going to run a little by tech based on some of the feedback I got from you guys. But do call your friends and let them know that this is some good review and some good questions going on. And we'd love to hear more from you. If you have any more requests, please do go ahead and let us know. The more we can get traction on this, the more, you know, we can do these in the future. So thank you so much for hanging out with me. Thank you as well to Nick. I do apologize for the tech issues, and we'll see you back here tomorrow at 3 p.m. Eastern.